All right, ladies and gentlemen, this is part two of DNA. It's all about making a protein. We also call it protein synthesis. So, as you guys probably remember or probably know, a lot of the things that we refer to as proteins are foods, so meats, cheeses, and fish. We're going to talk about it more in terms of its functions and what they actually do, and then how we make them. This is actually what a protein structure looks like. This is hemoglobin that we've talked about before. It's got its four different subunits, one of these quaternary structures. So this is a protein, even though we think about it like this. All right, so why are we even getting into talking about proteins? Well, all the proteins have a specific function, and in order for those proteins to be made, uh, a certain switch has to be turned on. And so what that switch is, they're called genes. So as we talked about before, this is a DNA helix, and a certain section of that gene or of that DNA is called a gene. And so these code, even though got it right here, code for a protein. So if that gene is turned on, like the light switch, it's going to start producing proteins. If it's not turned on, it won't be producing any proteins. And we'll see how that happens in a little bit. Uh, so what is a protein? Now that we've learned a gene, all it is, we know that before. It's a bunch of amino acids being held together by various bonds and structures. So we have that primary with the amino acids turning into a polypeptide. We have the secondary where you have those alpha and beta sheets. We have the tertiary where it continues to fold, and then the quaternary where it's multiple polypeptides joined together. All right, so just as a quick little review, RNA, we have this single strand of nucleic acid. The whole function of this RNA, well, the one that we're going to be talking about, which we call messenger RNA. There's three types that we'll see in a second, but this is the one we're going to focus on. It's the link between DNA and protein synthesis. So the DNA has the message, those genes on what protein to make, and then the RNA has to run that message to the ribosome where proteins are made. Okay. So detail things like we talked about in biomolecules, single-stranded, single helix, which means that it's just got this one coil, and it uses uracil, not thiamine. Oops, sorry, you might want to write that down, pause it if you need to. Like I was saying, there's three types. We have messenger RNA. This is the one that we're going to refer to the most. This next one, transfer RNA, we're going to talk about this one next time. And then this ribosomal unit, RNA, all you have to think about is it makes up the ribosome. And this is where proteins are made. Okay, so the location. So, like I was saying, the DNA message is going to be taken to the ribosome by the messenger RNA, and then the transfer RNA is going to bring those amino acids to the ribosome. And this is called translation, which we will see next time. So gene expression, which means making the protein, is made up of two main steps. This is the one that we're going to deal with today, transcription, where we're going to take DNA and we're going to change it into RNA. And then next day we're going to do translation, which is taking that RNA and then moving it to the ribosome where it's going to tell what the sequence of amino acids is going to be. So you pause that and write that down. Okay, so what is transcription? So what happens, let me just put these up and I'll pull up a picture. You guys have this on your handout, so just make sure that you're following along here because it's a little bit tricky, but it's really similar to replication. Okay, so what's going to happen is one strand of the DNA is going to get copied into RNA, just like the one strand of DNA on both sides got copied into DNA in replication. So it's a little bit different. So first of all, instead of DNA polymerase, it's RNA polymerase, and what it's going to do, it's going to bind to the promoter region. And all this is an area, oh, I guess it's right here, an area of DNA that contains special nucleotides, and those special nucleotides are going to tell it to start gene expression. So start transcription. Okay? And then what's going to happen, instead of DNA nucleotides, they're going to be RNA nucleotides. So you can see here's the RNA nucleotides coming to be joined in a complementary fashion with the change where uracil is used instead of thiamine over here. Okay, and same thing, they're complementary fashion. What's going to happen at the end though is this RNA strand leaves. So it detaches from the DNA strand here, and it leaves and then goes through the nuclear pore and into the cytoplasm. And then the DNA just totally recovers. So recoils up. So it just opens it up and gets copied and then coils back up like nothing happened. So once this RNA strand, so we have, all of our different bases, it still has that sugar phosphate backbone. So U, A, T, oops, wouldn't be T, let's do another A, G, C, C. So if this is our RNA strand, it's not completely done once we do transcription. It has to be processed. So what that means, and there's a picture in your notes about this too, is you add a guanine cap and then a polyadenosine tail. So that's like adenine and ribose. Adenine, ribose sugar. So that's like the ATP, adenosine triphosphate. It gives it an adenosine tail. And then what happens is any of the stuff that we don't need, because sometimes just because we copy it, it doesn't mean it's useful, will be cut out. They're called introns, so not useful. It might be more useful for another protein that they're making. So the functions with no function are cut out, and then what's left behind are the exons. So if you look here, so here's our DNA. It gets transcribed into RNA. We have to put on our guanine cap and our polyadenosine tail, and then we cut out any of the sections that we don't actually need. Okay? So what do you need to know? A little relationship between the DNA and the genes and how the amino acids make up proteins. Differences between DNA and RNA, which should be just a review. This is the main ones, obviously, the steps of transcription. So here's just a little recap, just a little visualization. You can go through this if you like. You might have to pause it because I'm not going to wait for it. So here's the promoter region. So it's a certain section of DNA. What's going to happen is when it zooms in, the RNA polymerase molecule is going to join and expand it. So it pretty much works as helicase because there's no helicase in this one. It starts coding, so here's your RNA coming out of it as it goes. And as you can see, here's what's happening. So each time it's only going off of the one DNA strand. It's complementary pairing. So your A's are going with the U's and the G's are going with the C's. And then you can see after it continues to fold back into DNA again. Once it gets to the end, it's a terminating sequence of DNA. It tells it to stop. And then finally, the RNA polymerase leaves. DNA is perfectly coiled up again, and you have an RNA strand that's ready to go to the ribosome to be changed into a protein. Okay, let me know if you have any questions. Please write down any questions you have because it's going to be very useful for our discussion group to start next day.